Okay, great. Thanks. And thanks, everyone. So what I'll do is I'll try to present some of the work that I've done, as Sikandar already gave a kind introduction. So I am presently working in the clinical trial side of the Novo Nordisk, where we are trying to understand how we can use, in different capacities, omics in clinical trials for drug discovery and decision making. But most of the work that I will present is majorly from my early side of the research that I've been pursuing at Novo Nordisk for the past few years. So my topic will be mostly covering how we are using omics to derive insights in early drug discovery, and then how some of it is following through the late stage clinical development. The talk will be primarily divided into three. So we'll just give you a flavor of our area. We are called development side. We are data science organization in the development side, and how we are using and leveraging data. There will be one use case of a multi-omics project where I have used a biobank in collaboration with our West Karolinska Institute, where we're trying to study atherosclerosis. And then we'll give some very small highlights of certain proof of concept studies in chronic diseases where we are using single cell and spatial for drug discovery and beyond. So we are a part of the data science organization in clinical development. As you can see, it has a large global footprint, and we have different expertise and skill sets, over 1,000 colleagues. And this footprint is very global at now. We have more than very diverse nationalities, and we are trying to use different flavors of data operations, ML operations, data science, and so on and so forth to help facilitate drug development. Our core belief and our core mission is to understand how in Novo Nordisk we can actually be a global healthcare leader by leveraging the power of data and analytics, and how data and data assets and data technologies and digital technologies can actually be a core enabler that can help us to change and defeat diabetes and other serious chronic diseases. But now I'll slowly shift towards the science. So what I have been studying over the past 12 to 15 years is basically, trying to understand the central dropdown molecular biology, where insults can happen at different processes before you generate a phenotype. And this I have been trying to study in different and various capacities using different types of omics data. And this is where I'm kind of giving an overall framework, how I've been able to do these kind of studies in different capacities of different projects. I've been very fortunate enough to collaborate with different departments, different therapy areas, where we have had access to biopsy, omics from biopsy data, omics from biofluids, and then clinical biobank informations from clinical registries. And what we have done is basically in collaboration with our stakeholders, be it from Novo Nordisk or academic collaboration, we have been able to generate different kinds of omics. What we did that is then we, depending on the question, we have tried to understand the different kind of molecular networks, both at the organ-specific level or even at the cellular levels, and try to close the gap between the genotype and phenotype relationship, which could help us to eventually propose some mechanisms, candidate drug targets, which we then take back to our molecular biologist and wet lab biologist, who will then start validating these through different series of checkpoints, so that we can eventually propose them as candidate drug targets. And this happens mostly in the early part of the research organization, not that the trials. So trial is a very, very late part. And this is what I have been doing majority in the last 6 years in Novo Nordisk before I shifted 4 months ago into a clinical trial setting. And this we have been doing so on and so forth in different kind of therapy areas. So as you can see, we work in diabetes, obesity, cardiovascular disease, of different types of cardiovascular disease, from heart failure to atherosclerosis, chronic kidney disease, then liver disease, so metabolic dysfunction with steatohepatitis, and rare disease. And here, what I'm trying to show is, if we now start putting them in the context of omics, how we can use it for target identification. And when I say target identification, I'm referring to candidate drug target identification, and how omics from different kinds of tissues that can be, biopsies, that can be biofluids, are taken together from different studies, different cohorts, and how we are leveraging them in an integrative fashion. Integrative fashion can be paired, and that can be non-paired, to elucidate different forms of evidence that can give us an overall evidence package in a human-centric manner, which can give us to understand how genes are associated with disease progression, what are the underlying pathways, if there are very specific molecular hubs, and if around those hubs and genes, do we have any kind of SNPs that are associated with our trait of interest, and what are the different kind of cell type association that we can find for a particular gene or a protein, and how well they are localized into the tissue. Taking all this integrated evidence using different kinds of statistical models, where possible, we also use, and I have been able to team up with some very um, uh, very highly skilled scientists, where we've been able to also use machine learning and deep learning where we could, to actually then come up with a list of pathways and networks that are either activated or deactivated into your disease of interest. Where we have clinical longitudinal measurements, we have been also able to identify things that are associated with progression. But that also means that it can be a huge list. So what we do is we further then prune them based on the biology and the hypothesis it's generating. And there we often come up with some tricks where we are either, depending on how big our findings are, and how complicated this information landscape is, we then use different techniques like knowledge graphs, where we can further prune these things. And eventually then we go for screening, and we try to understand, can we actually build something that is based on gene editing, or that is based on the small molecular peptides, which can give us some sort of cell type specific potential drug targets. This is the overall schematic when we think of how system-driven target identification works at our age, across beyond different organs and beyond therapeutic areas. Now I will shift gears to give the first example of one of the multi-omics project where I've been involved for the past couple of years. And this has been a collaboration with Karolinska Institute, where we are trying to study atherosclerosis in type 2 diabetic and non-type 2 diabetic background of patients, and try to understand plaque instability. There's a high unmet need. There's not a lot of medications out there, and it is predominantly a symptomatic condition. And it is one of the having high morbidity and mortality. As you can see, it's over 370,000 annual deaths in the Western world. And there are multiple risk factors, which kind of makes it much more associated with poor prognosis and poor quality of life. And type 2 diabetes being one of them, obesity, physical activity, and eating saturated fats becomes one of them. But what we are trying to understand through this project is basically, how does the different types of vulnerable plaque happens, and how that kind of proceeds to atherosclerosis, and then eventually with any kind of chronic heart disease. And with type 2 background, does we see similar patterns, or is it something more that we can learn out of it? As you can see on the right, let me try to see if I can create a pointer. Yeah, is my pointer visible? If it's visible, then I'll just point it. So, and here what you see are the different kind of stages of how plaque actually happens. What are the different proteins and inflammatory cytokines that are involved in terms of the mechanisms of how inflammation and cellular adhesion drives the plaque destabilization. But to understand the molecular events, what we have done is we have got access to these biobank called BIKE from Karolinska Institute, which had actually secured over 1,000 patients and they were trying to understand. This multi-center observational study was trying to evaluate the cause of long-term mortality in carotid endarterectomies. So what we were able to do is, in collaboration, until these clinical demographics were available until the end of the last year, we were able to actually profile at different levels around 40 to 60% of these patients, which is around 1,000 patients who were followed up for 10 years. And we were able to generate different types of omics, as you can see in the right-hand side. And the important part is we were able to profile we were able to do SNP arrays, we were able to do RNA-seq, and we were able to do proteomics with Olink, and then metabolomics with Metabolome. The important part is it was a very unique setting where we actually had the plaque drawn from the neck of the patients, and then we were able to also get the blood around the plaque region and the peripheral blood. Now, this becomes very unique when you have profiled so many different things where you have local blood, you have peripheral blood, and you have the tissue as well, and you have different types of omics. So it becomes a multifactorial problem. So we were trying to understand how can these kind of information add value to what we know about around atherosclerosis in the current landscape of the drug discovery. And multiomics in cardiovascular disease is also growing. As we see, we have learned a lot from the blood but over the years. But then, if we are trying to understand the deposits into the blood from the tissue, I think it becomes important that we also understand the tissue remodeling that is actually happening. And as you can see, there was a very nice review that came out last year, which was also trying to say like if how multiomics can be useful to study cardiovascular diseases, taking into account the cell, tissue, organ complexity, and also beyond just taking blood, right? How we can actually leverage the power of different omics with combining with the tissue context. And this can be done in different capacities. There are different ways to integrate multiomics data. One can be early, one can be intermediate, and one can be late. When I say late is you do all the analysis separately and then integrate in terms of certain networks. But at the end of the day, you are building up an interactive model with these different modalities or omics type and try to then derive insights. And these insights can be patient phenotyping, biomarkers, drug targets, molecular mechanisms, and then signaling hubs. So this is what we were trying to understand now, having this kind of data. How can we do, tick some of these hypothesis generation? Having said that, then this project aim was trying to understand what are the mechanisms can we identify for atherosclerotic plaque instability in these cohort where we had transcriptomics from blood and plaque, proteomics and targeted metabolics from blood, and we had at this moment we only had the information of a few clinical covariates, and we had labels where we know which patients or individuals were characterized as either symptomatic and asymptomatic with and without diabetes. And then we were trying to see, can we then create an analytical framework at our end, how we can actually integrate all this information together for a disease understanding context. We were particularly interested for this project at the time to understand if we integrate all these omics, can we study what are the mechanisms that are being contributing to the disease, so atherosclerosis as well as type 2 diabetes in the peripheral side of the disease, in the local side of the disease, are there anything that is shared, and how some of these feed into an hypothesis generation, if some of these are driven by inflammation, if some of these are driven by smooth muscle cells phenotype in terms of the cell types, or if something is endothelial cell dysfunction, which can eventually help us to drive targets. We also had an ambition we still have an ambition of building the biomarkers. However, at that time when we majorly work on this project last year, we did not have the entire clinical data. Right now, we just have started getting access to the harmonized clinical data with the follow-up. So we're also trying to touch the aspects of that as well. So here is an overall schematic representation of how we are using this data. I'm sorry, this is a pretty busy slide, but I'll try to walk over briefly. So what we did at this end was actually taking the transcriptome, the proteome, and the metabolome. I was not leading the part of the genetics one because the genetics one was being led by our colleague in the genetics department. My task was to work with a very talented postdoc in Karolinska Institute to actually make sure that we do systematic analysis of the other three types of omics together, integrate them across and do a bunch of different analysis. Here we are primarily looking into different sites across the symptomatic and asymptomatic and type 2 and non-type 2, and try to understand what are the molecular mechanisms that can eventually go for a drug discovery event. So what I did was we kind of picked up the method which is a part of these mixomics package called Diablo that we used here. We systematically looked into it and tried to understand. So it's a feature selection method which is supervised, and it helps you to actually integrate, so vertically integrate omics data when you have same number of observations, and that can actually then be used for any kind of biomarker or target discovery. So what you see here, we use them to actually find what are our targets, and then made some clinical association with some of the information that we had access at the baseline. and then further pruned them, looked into those genes and proteins and transcripts into the cell type, assuming for the proteins that we have a counterpart of what is happening at the transcriptional level, and then try to understand which can be interesting from the candidate drug discovery perspective, followed by a validation and screen. So we have now been also validating some of these results that we have generated from here. Having said that, I'll just give a very small technical briefing. In doing so, based on our experience, we did not want to just use the method Diablo as on its own. We wanted to do, because we didn't have a benchmark of the settings into the design of this method and how this should be used, what are the different design metrics one needs to use, what is the overall feature levels that should be a starting point, number of components that needs to actually capture the variance, but also show you the correlation. So we kind of systematically approach this. So here what it's showing is what are the different technical considerations that were taken when these comparisons were done, what were the different design metrics, and the ones which are bold highlighted is the one that made downstream for any kind of hypothesis understanding, let's put it like that. And then what we did was also when the initial optimization was being done for the Diablo, we also performed 10-fold cross validations for those optimal features. And finally, once you have these analytes that comes out of these feature selections, only using the ones that have a correlation coefficient around 50% were thought for any kind of downstream analysis. Here is a brief and insight into some of the results that kind of validates how this cohort is, whether it's capturing some interesting thing that we know, and then from there also trying to develop what are the things that are new. So what we found out from this cohort on the left-hand side is showing a few of the networks that were found in the peripheral and the local and the peripheral plasma integration. We were able to find during network analysis, both correlation and evidence, some networks that are associated with tumor necrosis factors and matrix remodeling, which is actually very popular in terms of studying atherosclerosis. The good part is we were able to find in this network the colors of indicate what kind of molecules they are. So yellow represents metabolite, proteins are in blue, and MRIs are in green. We were able to find very interesting ones that have also a literature and a positive control. So IL-6, which is actually there are evidences around its relevance in atherosclerosis and plaque stability, we're able to find them in our network. So which kind of makes comes out as a positive control that this cohort is actually capturing key markers as well. And then when we were trying to understand if there are any similarities at the level of proteins between the type 2 analysis and the symptomatic and asymptomatic events, we were able to also find key proteins that are associated and very well established in the field in terms of disease signatures relevant for atherosclerosis. Some of them are highlighted here. And then the last part, which also made us a bit more, let's say, comfortable and actually interrogating more around this part is what were the different kinds of pathways that we can find. One thing is finding the genes and proteins and transcripts, the networks and the interactome. The other part is also trying to say, do these findings break up into certain categories of pathways, both in the peripheral and the local, that are meaningful in the context of atherosclerosis. And we did find a lot of different cytokine and complement pathways that are associated with symptomatic type 2 atherosclerosis highlighted here. And these kind of helps us to say, okay, let's take the next steps. Here I'm just highlighting one of the proteins that is associated with endothelial dysfunction. When we take a deeper look, which is the FABB, it's a fatty acid binding protein, there have been quite a few literature around the potential of it as a biomarker, both in type 2 diabetes. There is also in heart failure. There are some very interesting recent prospective omics cohort analysis in heart failure, which shows this is an interesting biomarker. And there is a link around it in diabetes as well as inflammatory activation and endothelial dysfunction. So what we did was when we started looking into how it is actually associated with the different backgrounds, different medication, we were able to understand what was happening around it when we are trying to slice and dice the data to make a much more meaningful context. And on the right-hand side, what we do was like, given one of our goals is to also find mechanisms and targets and proteins associated with endothelial dysfunction, we try to go back to some publicly available single-cell data from the plaque view here, which kind of cell type overrepresents these molecules. So this is one of the finding, but we are right now assessing, overall like the number of targets and the mechanisms that we found out in this to see which can be eventually proposed as drug target. But this is just one cohort, right? So we wanted to make sure that with this study, we were able to establish a multi-omics framework that can help us to study symptomatic type 2 atherosclerotic patients, obtain some high confidence pathways and networks, and we are trying to that are relevant both in atherosclerosis and plaque biology. And then we were able to understand how some of these, proteins, uh, transcripts and metabolites actually talk to each other and converge into some meaningful pathways that can not only help us to study atherosclerosis, but the symptomatic and asymptomatic events, but also what is the contribution that is actually happening from local to peripheral site of the disease progression. One of the reason why we have been interested, or rather I have been interested in driving the multiomics, is I'm trying to more deviate out from the one gene hypothesis. When we talk of central molecular dogma of biology, we always try to understand something that is, having a more consolidated view rather than one gene hypothesis. So that's the reason, these kind of multiomics comes into picture for us. Presently, our colleagues in the genetics team are running the genetics causal analysis around these data sets and also the targets. And we are also running multiple survival models with outcome data for the phenotypic traits that have been measured. We have now access to the or let's say permission to run these models on the clinical data, outcome data that have been collated for the last 10 years on these patients. And we have actually been running multiple stages of screening and validation. Initially, we found around 25 let's say high confident and meaningful targets that we screened, and then we are running a bit more semi-throughput CRISPR around the over 100 targets. That is the overall. Having said that, those things are being run in conjunction with our other cohorts where we are able to see some of the same findings being replicated across the other cohorts, because we don't want this to be a one cohort or one shot wonder. We want to make sure that the more these cohorts are a part of a bigger framework where we have multiple similar cohorts to actually add more confidence. And we have presented this work last year in ESD, and we are also kind of writing a manuscript, but it will take some time for us to digest everything and put it out in a meaningful manner in a manuscript. Yes, now I will shift some gears. What I will be doing is shifting from bulk multiomics and tissue profiling to more on the single cell side. So we know that single cell is becoming very popular in the last and has matured a lot. And what you see here is basically how single cell disease atlases are actually aiding in drug discovery. They promise to actually be very impactful, both from the drug discovery, target discovery, or screening, or even repurposing purposes. And right now, there are multiple examples like human cell atlas, human tumor atlas network, then human HubMap, or the kidney precision medicine project, where these disease atlases are being generated across different academic and industry partners. And these projects, at least the human cell atlas, were held by Professor Shah Teichmann and Professor Aviv Rejeb, who have been pioneers in leading these efforts over the last few years. So having said that, what we are also trying to understand is, well, these are great. We know that human body is, composed of trillions of cells. How can we actually leverage some of this information at our end, but we can build our own disease atlases for the kind of diseases that we are interested in Novo Nordisk, which are primarily cardiometabolic chronic diseases. And we are doing it using the single cell technology. Primarily, our work has been very much focused with the 10x, but we have also tried to tap into some other technologies as well. And here is a very small schematic of how we see that single cell can be potentially helpful. We are kind of piggybacking on the paper that was published by AstraZeneca, where they tried to understand, when we want to do effective target discovery, we need to really understand how well the target fits into the context of the tissue, the safety, the right patient population, and what is the commercial potential. And we think a couple of years ago, a few of us, me, Simon, and my former manager, we wrote a white paper where we're trying to understand how does single cell actually fill into that context. Like if you're able to perform some of these, if not all of them, into the context of single cell. So we were able to see that if not all the 5 Rs, it was able to actually address 3 of them. And this is where we started different kinds of projects a couple of years ago using single cell to build our own disease atlases. However, this field can be very tricky and complex given how the data is generated, what the data looks like. I think most of you are very familiar with. There are quite a lot of challenges with single cell data that can be, classified under or categorized under technical, in terms of how the amplification bias can happen, how the RNA input is, what is the normalization, if there's a dropout, what is the QC. Then there can be something with the methodological aspects in terms of the library preparation, what kind of cell selection we are doing, dissociation analysis, and so on and so forth. And then there can be biological, which is more like the variability around cell, the heterogeneity around the tissue context, and then splicing and gene isoforms. Given how these data sets are being generated. But the good part is, there were very nice papers that was published by a very great group of scientists who put out these, what are the different kind of challenges that we see in the data science of single cell. And this was something that we also kind of looked into a lot while we were developing our our workflows and projects around how we can leverage single cell more in the applied context of drug discovery. The good part is, as over the years, quite a few of these things are already addressed that were highlighted in this paper, if not all. And in doing those, we were also actually able to kind of make a much more strides at our end, taking a cue from these kind of developments that have happened in the single cell, where we were trying to develop our own atlases across the healthy and disease patients' data. So one of the example is our own cardiorenal disease single cell atlases that we are trying to develop in collaboration with Unilink Aachen. We have a very talented postdoc, Thore Blackpool, with Sikandar and Professor Rafael Kraman, where we are trying to kind of generate from a publicly driven, so publicly available data that is there in the around chronic kidney disease, around atherosclerosis, and around heart failure. We're trying to take all of them, run the analysis from scratch, try to harmonize and standardize this data, and then try to build up the unified single cell atlases. And then these can be then eventually used for drug target discovery, any kind of association with the clinical manifestations, cell-cell communication, and different kinds of, downstream secondary and tertiary analysis that can help us with drug discovery. Here is a very small part of this work that I'm showing. I think I would really encourage everybody to go into the preprint that's already out there. This work has been under review. A bit of a glimpse I'm showing here of this work, where we are actually trying to understand how we can first integrate all these atherosclerotic data that are publicly available. Understand what is how does everything can be harmonized, how we can annotate these cell types, and we have used both automated method, plus we have also benchmarked a couple of methods for the integration, and then we have worked in collaboration with molecular biologists to see how our cell type annotation are kind of congruent and make sense. And then we try to understand the spatial diversity. What here I'm showing is basically what was the overall number of cell types that we were able to annotate. And then when we perform different kind of analysis in terms of the genes that were changing between control and atherosclerosis, what was the overall number of genes that we got, and what are the different pathways that we found out. Interestingly, there were quite of pathways that were related to the immune system, related to angiogenesis. But what caught our attention was this small blurb of pathways that we see into the pericyte, if it's visible to you guys. We saw a lot of things happening at the pericytes, and that kind of piqued our interest a lot, other than what is happening in the vascular smooth muscle cell. And we have been trying to understand that a bit more and address that part, like how pericytes is associated with immune modulation, differentiation, and angiogenesis, and how the single cell, in conjunction with the spatial data that was also available in this collaboration, was able to help us understand the trajectory of migration of vascular smooth muscle cell into more fibrogenetic cell type, cell phenotype. Let's put it like that. Having said that, this is a lot of work. I'm not doing a justice in in putting up the highlights, so I would kind of request everybody who are following these kind of work or in doing atherosclerosis research to check out our publication. Right now, it's still a preprint. We are addressing some of the reviewer comments. And then, as a part of this huge collaboration, we are also now developing a heart failure atlas. This is combining around more than 6 studies, around nearly around 200 patient samples, 1.8 million cells. This is currently a very, very large effort. This is still ongoing. We are still right now teasing out the different technical aspects before we can make a lot of inference right now. Here is a very small schematic of overall what we have done in terms of the integration and some of the cell type labels that we have been able to annotate. But this work has a lot to be ongoing so far, because there's a lot more that we think that we can actually generate in terms of hypotheses and findings from this work. Now I'll be shifting gears a bit more into the spatial. I said that in the previous work we had some spatial data, but I've not forayed much into what capacity we are using it. So we think, and I also personally think, with our talented group of scientists at Novo, that spatial transcriptomics has a lot to offer in terms of drug discovery. We think it's a very valuable technology in terms of doing in situ hybridization around the transcripts at a scale. And if we want to pinpoint different subcellular location across healthy and the diseased tissues, understand the tissue microenvironment and some of the cellular dynamics, we can actually use this and explore and exploit this technology a lot more. And we can also infer and validate our findings when we're doing these ligand receptor analysis, where we can actually find now how known ligand receptors are actually communicating between different cell types from the single cell data, how well we can actually see those being distributed in the tissue neighbors on a slide. So this is why we think it becomes a very integral part in the drug target discovery and validation. And I will give you a few glimpses of some of the works we have done, very few fast glimpse. So what we did was we actually broke down this work in 3 different short works. First, we tried to understand, when we are internally generating single cell and spatial data at our end from human cohorts, what is these are primarily 10x Visium slides. 10x has its own problems in terms of how these spots are. They are not single cell level, they are spot level, which might be 10 to 30 cells or nuclei at best. So which means that we need to run into a deconvolution project. And for our own data, we wanted to understand which are the deconvolution methods that we can use. So we did run a short study where we tried to systematically evaluate 3 methods, as you can see here: cell to location, RCTD, and the DWLS. And we were able to figure out, using both statistical models, but also histopathologist congruency validation way, to see how well these methods are able to actually identify with highest accuracy or deconvolute these some well-known cell types, but also somewhat that are rare. And we found that cell to location and RCTD were the ones that were having the highest accuracy in the tissue of atherosclerosis, as well as the chronic kidney diseases, which are actually not very well established in the current benchmarks that have been done. That was the motivation for us. And then we started using cell to location, which is much more flexible in terms of what can be done at our end for different other projects. And the other, as I say, other projects, is now we were trying to push our ourselves to say, okay, now we know which method works in terms of the deconvolution. Can we then start using that plus start making some biological hypothesis generation with in combining both single cell information and the spatial. And this is a project where another master student actually did a thesis around, where we were trying to study in two different chronic disease. One is CKD and the other is liver disease MASH, metabolic reprogramming across the different stages of the disease, as you can see. And we used, as a proof of study, single cell data and spatial data from publicly and privately available cohorts, and try to understand what are the ligand receptor that are actually changed between from early to the late stage of the disease, and what are the cell types that are actually changing and making those changes, and how well some of them are represented in the slides. And here is the overall workflow of how we were actually taking different methods that have been published, and some of them we have also internally evaluated, how well they are actually helping us to combine this information, which can actually say be instrumental in terms of deriving targets and hypotheses around targets, and how well some of those targets are validated in a spatial slide context. And this work was presented also in ISMB last year, and recently we have been able to get this work also accepted in a paper. This will be coming out soon in a few weeks and available online. And here is some results around that. What we are trying to show here is basically when you have single cell data, when you have spatial data, you can actually find composition clusters and then try to understand and infer ligand receptors across different cell types, and then actually try to see what are these ligand receptors, how they are actually changing across the different state of the disease. And now if you want to map them back to the slides, if you are very well aware of the different fibrotic and the metabolic markers, you can actually then juxtapose the expression of these ligands and receptors on them to actually make much more improved understanding of what is happening at the target level, which we think is actually what very, very valuable from the target discovery and the validation perspective. I'm in the last part of my slides. I'll not take much more time so that we can have some time for the questions. But all these examples that I've shown are majorly from cross-sectional studies, right? So what we do in a cross-sectional study is we try to, profile different groups, and we, as represented in the left-hand side of these, diagram, and we try to evaluate how they are actually compared at the same time. Even though we say that, we are actually looking into different stages of the disease, but ideally they are not always longitudinal in nature in terms of how we profile. And there are caveats in that, because it's not that easy to profile certain complex tissues, right? But what we want to study when we are doing drug discovery, when we're doing biomarker discovery, is longitudinal design, where we want to study the same group compared over time. And this is where we think that a lot of things have its limitations when we do these through observational cross-sectional cohorts, even though they are valuable in terms of hypothesis generation, but they have certain limitations. And this is where we are thinking that how we can actually now do these things in a more longitudinal setup. We are getting access to prospective cohorts from the early research space, but we are also trying to see, do we have, from the trial perspective, in generating these kind of study designs or not? And we're trying to do this both from the bulk omics, so proteomics, metabolomics level, but also at the single cell level. And one of the example where we are trying to push the frontiers in the single cell level in the clinical trials is this seminal study where we are trying to understand in type 2 and chronic disease patients how semaglutide works. And it's an outcome trial that we are doing, and we are trying to understand how the drug works, but the mechanism is being done. The understanding of the mechanism of the drug is being done with a very short mechanistic trial called Remodel, where we are trying to have patients with entry and exit biopsies before and after the treatment, and after the treatment of 52 weeks, so 1 year. And then we are actually taking these biopsies and trying to, profile both the single nuke RNA-seq as well as the spatial, to really understand what are the genes, pathways, and the programs that are actually changing with the drug treatment. And this is our way to also really understand that how some of these technologies, which have been very instrumental in the early space, are also valuable and beneficial in the late stage of the clinical trials as well, when we're trying to understand drug mechanism of action. Yes, and this is the slide where I'm trying to also say that as a computational biologist, most of my work that I've shown today is applied. We have not developed any method. We have developed, frameworks and, validated something. But there's a bug in me which also tries to understand, these data sets are very amazing. They're highly dimensional. Can I actually team up and learn from, very talented mathematicians and machine learning scientists, if we can develop some machine learning models with certain problems that are still there with high dimensional data. And here are some of the works that we have been able to publish. And this is a collaboration that I do outside of my current job with Indian Statistical Institute in the machine learning unit there, where we have been actually basically with very limited funding, working with problems of publicly available omics data, and identify the mostly developing machine learning models, primarily autoencoded-based models, to understand the non-linearity when you try to integrate different types of data. We are interested in bulk and single-cell multi-omics methods, latent embedding methods, and then a bit of cell type annotation. Most of our works have been at the level of feature extraction, and now we are slowly trying to understand if we can do some feature selection parts as well or not. But these are not at all in the space of drug discovery. They are very, very in the early basic research space of when machine learning is concerned. And finally, I come to my last slide. I think that our future of drug discovery and drug development is slowly moving towards cell-based interceptive medicine. It's an interesting time. We are generating a lot of data that is through different consortia, different public and private initiative, to study various types of diseases, be it cancer, be it chronic cardiovascular diseases, or any kind of cardiometabolic diseases. But at the end of the day, we are getting better and better to really understand, can we actually intervene through in terms of the cell phenotype or cell-based endophenotype. And this is where I think there's a huge playground for single-cell multiomics, as we see. And this was a very nice visionary, let's say, review that was published by Lifetime from a group a few years ago, where and I think it holds true right now with all these amazing publications that are coming out from different consortia initiatives as well. That having said that, we are in a great space to generate some very interesting data. This also provides a huge playground for predictive modeling, be it statistical models or machine learning, and then eventually also having a scope for taking these learnings together and then try to build more patient-derived experimental disease models. However, we need to be mindful that all these are predictions. If not well validated, there will be a lot of uncertainty around this, because some of the technologies we are using, even if they are full matured, they have their own limitations in terms of the chemistry and dissociation protocols, especially when single cells is concerned. And yes, here I would like to stop my talk, and I would like to acknowledge all the listeners and my colleagues at Novo, my collaborators, all my mentors, and the computational biology community and the applied statistical community. And I would try to now understand how I can stop my sharing my screen, and I can take up questions. Yes.
Thanks, Vivek. It was really insightful. I have a few questions, but I will just wait for a few seconds to give audience a chance to ask some questions.
If you click the share button again, it will turn off your screen share.
Okay. Is that okay now? Yeah, okay, great. Yes, I hope it was not super rushed. I kind of put a lot of information, but the point was to put how we are trying to think about these type of omics for drug discovery and how it feeds to some of the translational side of the late stage. But please feel free to ask me questions. I'll try to answer to the best of my abilities.
So, Vivek, I have a question regarding, let's say, two major things that you mentioned. You said that you have data access from biopsies and from biofluids. um, how important is it to have, let's say, non-invasive data together with the tissue of interest to think of, let's say, translational aspects of such projects?
That's a great question. So I have mostly shown today things that are pertaining to cardiovascular. A part of my heavy work has been chronic kidney disease, and there we have been trying to, we have done a lot of strides with, invasive biopsies and when omics is concerned. But the overarching also goal is to see how we can actually use non-invasive materials. So that becomes very, very important in terms of the biomarker discovery, how we want to, classify or say categorize patients. Having said that, biofluids, if not taken sequentially, can be very tricky in terms of, these kind of aspects of biomarker identification. So we have to kind of, leverage a trade-off in terms of what is our overall objective and the goals. In certain parts, we believe that biopsy is still very relevant. In certain studies and projects, we think that we can still do things without biopsies, and biofluids can be enough as long as we have sequential sampling available. So in terms of, let's say certain CV diseases, when we are trying to profile omics, we try to see if we have time points available there. And then if not, then we will go with cross-sectional biopsies. But it all depends, like if we are seeing something that is tissue driven, then we have to go back to the resource of the invasive manner. I hope that answers partly your question.
Oh, that sounds great. Yeah, thank you very much. So we have a question here in the chat. What are your thoughts on identifying distinct cell states rather than cell types in single cell and spatial data?
That's actually a great question. This has been something that has bothered me a lot in the last few years, personally, with all the amazing methods that have been, but also like I've been part of developing a method at our end as well. I think the cell state is a very, very tricky thing. At least in my experience, I think these atlases might help us to an extent, how we can actually build these cell states in terms of the continuum. Cell types is a very broad categorical level, but I think it's very tricky if you don't have, longitudinal sampling to really make an impact in terms of taking a cells, defining a cell state. I'm not saying we cannot do it entirely if we have enough depth in the data, if we are able to find pattern of transcripts that are, defining those continuum. But personally, I feel it's very, very tricky. It's very important to say, because all we are trying to define is our let's say our battle is against the function of a time, and these cell states are representative of that in terms of the disease status. So, but I still have my let's say reservations of how we define these cell states and how well we define these cell states. In some cases we do, but I think moving forward, the more and more data we get, and the more and more some of these things we can correlate with some of the clinical information, we will get a better granularity on that. And I know that some of these methods which have been developed right now from the foundational model perspectives, people are trying to get a bit better handle of those as well, using NLP based methods or language models. I think it will get better, but I still am not fully confident that we are fully certain around the type of cell states that we define. That's why what we do is basically, when we define something on the cell states, we go back to the lab and do in vitro test to make sure that we are able to perturb and see things what we are claiming from the high throughput data.
Okay, thanks, Vivek. One more question around, you mentioned GWAS, like SNPs and single cell data. So for, let's say, translational work, where you're coming from, let's say, not just an academic point of view, but also from let's say, company point of view, what is more important, GWAS or single cell, or a combination of both?
I cannot speak for the company. I can speak for myself. I think I don't see one single technology as a person. The personal opinion, I don't see one single technology is going to solve drug discovery or medicine or biomarker. It's impossible to do that. And that's why we are studying central molecular dogma, right? The insult can happen, and as we are born, and that kind of cascades through recruitments of specific environmental factors, which will then, impinge your transcription and translation, and then giving rise to a specific phenotype. So you will need all of these together in combination in different capacities. So of course, if we're talking about the landscape of somatic cancers, then it's all mutations. But if you're talking about chronic diseases, evolution of the diseases, I think it cannot be answered by one single technology. That's my personal opinion over the years, based on the experience, both success and failures, majorly failures. Let's put it like that.
Sounds great. Then there's a question about collaborative opportunities at Novo Nordisk. So you mentioned a lot of collaborations in the talk. What are some systematic ways to collaborate with Novo?
No, that's a great question. So we have funding mechanisms and academic industry collaborations called Novo Star. Please look into Novo website. There are different kind of PhD and postdoctoral programs that are available there, where we kind of collaborate a lot with different academic labs. We have to write proposals jointly with an academic PI, and then we have to contest it at the different time deadlines. And then if it gets selected, then only those collaborations start initiated. There are other funding mechanisms, but I don't remember the back of my head right now. But these are some of the ways where we do kind of industry PhD and postdoc programs in terms of the collaboration. I see there's one question around diagnostics space, if I understood correctly. If that is so, I would just say that we are still very early. We are a drug discovery company working mostly in terms of translational science, in terms of drug discovery. We are not there yet in terms of diagnostic per se yet with the omics side. Eventually, maybe in future we will, but it's too early for us to get into that space.
Sounds good. Maybe one more technical question from my side, and then hopefully we can get some more questions, or if not, then we wrap up. So the question was around integration methods or approaches that you mentioned before, early, late, and intermediate probably. Generally, when we look at, let's say, single cell or any omics data, at least in my experience, it's not the case that we get multiple omics from the same sample. So let's say if you have metabolomics or proteomics or single cell data, what are your strategies to integrate, let's say, data which is different modalities and coming from different samples, but trying to answer the same question?
Yeah, so this is where I think the late integration comes into picture. If you're doing it with gene programs or gene regulatory networks, right? That's where you can actually do certain integration. Having said that, there are now some technologies, like I have extensively worked outside of Novo with a technology called CiteSeq, where you can jointly profile surface epitopes in terms of the proteins as well as mRNAs. I think that is becoming very popular. You have now these multi-omics, which is single cell and ATAC-seq. So these are there, which is also kind of using. So they are kind of bimodal. Let's put it, not proper multi-omics in if it's 3. So these are there. Otherwise, it's mostly gene programs. We have integration. So network interactome integration that kind of has been working so far at our end. This is one of the ways with we kind of integrate these different kind of information.
Sounds great. So maybe just one final question out of curiosity: what are the techniques that you think? You said that in the spatial world, this could be a major player. What are the techniques that you're most excited about in the spatial world?
Wow, that's a very interesting and tough question as well. Well, I mean, I am more looking forward to single cell resolution of spatial omics, both at the transcript level as the protein level. I think the protein is something from a drug discovery point of view we are more interested. Transcripts is great, but then that's a huge leap of faith assumption that things will translate. And if we are able to get a technology that can profile at the single cell level, tissue, both the transcript, the protein level, that will be super exciting and super interesting from the drug discovery point of view. IHC and IH are great, but they have their own limitations. So if you can scale it up in a much better way, then I think it helps the drug discovery industry as well a lot. And it's just not the industry. I mean, it will help in terms of hypothesis generation as well as confirmation as well from both academic side and industry side of the research.